डीएनए मध्ये तयार होणार आहे त्या origin point ला, that point which is called O, origin point or ODI also, that point DNA molecule cut, and that enzyme which is endonuclease enzyme, that enzyme which cut the DNA molecules here, and then that is DNA helicase enzyme. These DNA helicase enzyme that unwind the DNA molecules. We know that is the DNA is the double helix structure, that is coiling structure, that is the double helix structure, and these double helix or these winding structure is unwind here or uncoiling here, that with the help of enzyme DNA helicase here, cut and unwind here, and these unwinding which again coiling and recoiling also takes place here, but this prevent by the SSBP, that is the single cell binding protein, that is the SSBP that holds the strand here. हे जे स्ट्रँड आहे हे स्ट्रँड हे पुन्हा रिकॉइलिंग होऊ नये, हे जे वाइंडिंग अनवाइंडिंग झालेलं आहे किंवा हे डबल हेलिक्स खुललेलं आहे ते पुन्हा रिकॉइलिंग होऊ नये किंवा अगेन एंड अगेन रिकॉइलिंग होण्याची शक्यता असते, ते होऊ नये त्याच्यासाठी मदत कोण करतात, दॅट इज द सिंगल सेल बाइंडिंग प्रोटीन. ते सिंगल सेल बाइंडिंग प्रोटीन हे डीएनए स्ट्रँडला स्ट्रेट पोझिशन मध्ये ठेवतात. देन the fifth step, that is the synthesizing of new strand. Synthesize of new strand. Here the fifth stage or fifth process, that is the synthesis of new strand. This synthesizing of new strand, DNA चा जो नवीन स्ट्रँड सिंथेसाइज होणार आहे तो मनाने डीएनए स्ट्रँड सिंथेसाइज होत नाही. इट हेल्प्स इन which is the RNA primer. RNA प्राइमरच्या मदतीने काय होतोय, नवीन स्ट्रँड हा काय होतोय, सिंथेसाइज होतो. RNA प्राइमरच्या मदतीशिवाय there is not possible the synthesis of new strand here. Here the third end and fifth end, here third end and fifth end, this DNA molecule is the anti-parallel, that is the fifth to third and third to fifth direction here. A DNA molecule which is the anti-parallel position, and the RNA primer bind to the third prime end here. It also called, that is the third prime here, that is RNA primer bind to the third prime end. Here also RNA prime bind to the third prime end. We discuss now the synthesis of new strand, and new strand we started from the synthesizing with the help of RNA primer. RNA primer which is the fragment of RNA. RNA primer which is the small fragment of RNA, and these RNA primer which synthesize with the help of DNA primase. These RNA primer which synthesize with the help of DNA primase here, RNA primer. And RNA primer always bind to the third end template here. RNA prime bind, it bind not the fifth end. It bind not the fifth end, it bind the third template strand here, and these RNA primer, हा RNA primer bind झाला की हा काय करतो, these RNA primers are attract to the nucleotides of the nucleoplasm. न्यूक्लियस मधल्या न्यूक्लियोप्लाझम मध्ये जे न्यूक्लियोटाइड्स विखुरलेले आहेत त्यांना अट्रॅक्ट करतो आणि तो काय करतो, त्यांना बाइंड करत जातो. RNA prime are bind, attract and then bind, and which these RNA primer are these newly strand here which is the template strand here, bind the hydrogen bond here, A to T, T to A, or G enzyme